Yehudit, I said I wanted to get into the stem cells. How did you move from immunology to stem cell research?
In fact, it wasn't that hard because, as I explained in the immune system, we were interested in epigenetics. In epigenetics for one, and then when I started my own lab, I was also interested in the phenomena of repression. So you have genes and you either activate them, and now we know that you can also repress them.
And most of it's repression, isn't it?
Right. But when I started to think about it and to work on that, in fact, a colleague of mine was telling me, why do you think active repression really occurs in mammalian? Are we similar to bacteria? And that was an intuition, in fact. So I looked for a system where I can study epigenetics and repression, and it will be meaningful. So I just moved to stem cell biology.
But, I mean, one of the characteristics of mammals and humans, as I gather, you have a different kind of system in which we, well, why don't you you can describe it far better than I'll ever be able to do it.
That's true. We have different type of repression. When we think about repression in our cells of the body, it's very different than what we just say about repression in bacteria. But there are commonalities, but with higher organisms, we have different additional levels of repression. So we have everything there. And it was of interest to me, what are the levels of repression that are taking place, and what is the role of this repression in stem cell biology. So this is how we started to study how you turn off genes that are important for the pluripotential phenotype of a stem cell. A stem cell can differentiate, an embryonal stem cell can differentiate to all tissue of the body, and for that it needs to turn off the program of his identity.
Oh, right, that's right. It's not only the other identities, but it's only.
So it turns off his identity and turns on the new fate that it's taking, and also turning off the fate of the other cells.
So it can't be a kidney if it's a heart, or so it has to turn off that you're not a stem cell anymore. You're not a heart cell. You're a kidney cell,
just a lymphocyte,
or whatever it is. So what particular aspect of that turning off did you work on?
So in the genes that we work at, the pluripotent genes, in fact, we worked on 3 different aspects. The first aspect is just turning the machinery off. So you have a machine that comes to the gene and starts to make RNA from it. So the first level of repression is telling the machine, stop working now. You are not going to go through the gene and make more RNA. So this is the first level. And then comes the second level. The second level, remember I told you about the envelope of the DNA?
Right.
So the envelope of the DNA has to change to an envelope that says, I'm a silenced gene. So you change the envelope. And only at the third stage, you methylate the DNA. So you add something to the DNA that says, this is really a light that says, no opening here. This is completely closed. So these are the 3 levels that we studied in turning off, in repression of pluripotent genes.
So the mechanics of turning off these genes to make turn an open, effectively a stem cell system is an open system with all kinds of possibilities, and it ends up creating all these closed systems. How is that related to, because you start doing work on cancer, how is it related to cancer?
It is
related. Which is a third field. You jump from stem cells to cancer now.
Because these pluripotent genes, some of them at least, that are so important for the self-renewal of the stem cells, that the stem cell knows how to renew itself, that its daughters and sons are going to be stem cells. This is the meaning of the self-renewal. So the genes that are important for the self-renewal are great for a stem cell, but if they are staying on and stay expressed, they are very, very dangerous.
That's a cancer cell.
Exactly.
That's what a cancer, the definition of a cancer cell.
Exactly. And so we showed that at least one of them that we worked closely on, is exactly what you just said. It's an oncogene, what we call a gene that creates.
Won't turn off.
Right, won't turn off, exactly.
I mean, that's the most exciting work I think you do here, and in your paper when I read that, here you find a mechanism that won't turn off the gene, and so you get a wild gene or a self-multiplying gene. That could help you in diagnosis, right? You can figure out, or have you figured? You're not to that level that you figured that out yet.
No, we did.
You
yeah,
have.
Because we looked for us to know that we are on the right track. The first thing that we did, together with a collaborator of mine, Dr. Eli Piekarski, who is a pathologist, we looked on a variety of tumors that we suspected that this particular self-renewing gene may serve as an oncogene. And we saw that in all of these particular set of tumors, these are tumors of the germ cells, the sex cells, all of them express this particular gene at a wild levels. So only after we knew that, we said, okay, let's really see whether mechanistically it does what we think it does. So we moved to a mouse system where we can manipulate the level of expression and show indeed that this is what happens.
So how does it come to diagnosis of the disease? How do you use it then?
We don't now pathologists know that tumors of this particular type, they express this particular gene.
So you can detect it, and
Sure,
there's a test,
sure.
and
Yeah,
so you.
it's an easy test.
So it means it's very early. Then you can start detecting.
Right, exactly. In fact, we show that you're absolutely at the point. We show that a state of tumorigenesis before it becomes a really malignant tumor, at the very preliminary stage of this particular tumor, it starts to express this particular gene. So it's really a very early event.
So even before you effectively have cancer, or know anybody has any sense you have cancer or you're sick or anything else, you can figure out they have cancer or pre-cancer or predo-cancer, whatever you want to call it. So is there a system now which you can cut off, or is that what you're working on?
Exactly what we're working on.
Yeah, so that's what you're working on, that you say, oh no, your switch we got to change your switching mechanism.
Right.
And how would you do that? Do you introduce a new amino acid? An enzyme? How do you do that?
If you know how you turn it off, then you can think how to modulate the signaling to turn it off, one thing. The other possibility is that you have to find which is the target of this particular gene. Okay? And if you figure out which is the target of the gene, then you turn the target off.
I
so
see.
this
So
is
that
a
it won't
cascade of
so
events.
it only has one effect, and then you stop it right there. And so it doesn't, so that's two different strategies, but you can work on both, I guess.
That's true.
If you do it both, you got a double.
That's right. That's right.
And that's your new work.
Yeah.
But I read your new work. I read a piece as another fourth area again. You're doing tissue regeneration, which is a total new area.
It's a total new area.
And okay, well, we'll take a break because I want to talk about it. But I also want to talk about my startling kind of a discovery that, for me, because I'm not a scientist, that there's a connection between the way we develop as, you know, from stem cells and all the good things, the embryos and all the things that come from, and cancer. But we'll save it for after the break.